Bonjour tout le monde, merci d'être venu assister à la présentation Québec-Océan d'aujourd'hui, donnée par Monsieur Eric Orenstein. Alors je vais passer à l'anglais pour présenter Eric. So Eric is a very new senior scientist at the National Oceanography Centre in the UK, in Southampton, southern UK. Eric got his PhD at the Scripps Institution of Oceanography when developing automated classification approaches to process in situ plankton image data. And I've personally met him just right after that for a postdoctorate internship at Villefranche in France, working on the same topic. And Eric did a tremendous job during right at the beginning of the pandemic and all the aftermath of that. So we actually met in person very rarely, and I'm glad to have him here. So Eric is currently has been a research engineer at the Monterey Bay Aquarium Research Institute in the US, and his work lives at the intersection of machine learning, ocean imaging, and marine ecology. Automated detection and classification models often fail in the context of rapidly fluctuating ocean ecosystems and conditions. So Eric spends a lot of time thinking about the root cause of that behavior and ways of ameliorating them to speed ecological analysis. So today his talk will be entitled Intelligent Autonomous Sampling of Marine Ecosystems. And that's it, Eric. Thank you.
Well, thank you, Fred. Thank you all for coming, both in person and online. I apologize, despite having spent that time that Fred referred to as a postdoc in Villefranche. I did not learn any French, so I have to give the presentation in English. And even if I did, it probably would have been insufficient to actually sound reasonably intelligent. So anyway, I am going to be telling you all a little bit about some of the work that I've done. This is mostly stuff that kind of spans my time both as a PhD student and now as a research engineer at the Monterey Bay Aquarium Research Institute. And as you'll see in this talk, some of the same themes kind of run through, both in terms of the type of organisms that we've been looking at, but also some of the engineering challenges that we've encountered along the way. But to start out with, and I probably don't need to tell most of you in this audience, that when we're doing sampling in the ocean, we're often asking very fundamental questions, relative to what some of our terrestrial colleagues might be thinking about. And so I think that in Emmett Duffy's new book on ocean ecology, he laid this out very nicely in the introduction, asking questions like, how does the environment influence the distribution and diversity of life in the sea? As we know, it's a very dynamic environment, changes things on very short spatial and temporal scales. And then how do the interactions between organisms change those patterns? So things eat each other, things move around, things encounter each other at different rates. And then thinking through things like, how does all this impact the flow of energy and matter in this whole big complicated ecosystem? But we can even take these questions and I think boil them down to even simpler ones, and ones that get at the core of why it is that we put things into the ocean to try and look at the biology there. Things like who is there and in what number. So just identifying the things that are there and counting them. Trying to figure out when those organisms that we're looking at are there and when they're not there. And then what are they doing? Are those numbers changing in time? And what might be impacting those changes? And finally, things like, how are the organisms interacting? And while these questions seem like they're pretty basic, they're in fact very, very difficult to actually ask and answer in the ocean environment. And a lot of that comes down to how dynamic that ecosystem is. So there's an enormous range of spatial and temporal scales that can impact the organisms that live in the sea. You can think about things at a very, very large scale, like when you look at these satellite images of changes off the coast of Massachusetts at these, large kilometer scales. And then you can get quite a bit smaller than that and look at things on the mesoscale, looking at these slicks of plankton along the surface that you see in this picture off the coast of San Diego, taken from a drone. So things that are more on the kilometer to meter scale. And of course, you can get quite a bit smaller than that, going all the way down to turbulent length scales, and all of that impacts who the organisms are that show up in a particular place, why they're there, how long they're there. But it's not just that there is a big range of physical changes that impact the ecosystem. It's also that the ecosystem is very, very big. So the scale, not just of the physics, but also of the space itself, impact how difficult it is to look at the system. So in this very, very rough schematic of the open ocean, we have at the very top, the surface. At the bottom there, beneath that line, the seafloor. There's a benthic boundary layer about 50 meters or so above the seafloor that has very different properties than the rest. And then the photic zone, so that small band where light penetrates. And then in the middle, you have that giant stretch of midwater. And in a space as big as this, the organisms that you might see there are rather sparse. And actually finding them can be a big challenge. And as a result of that, given that we have this midwater environment that represents about 93% of the habitable volume on the planet, about 30 to 60% of the animals that live there are totally unknown to science. We've never seen them before, so even just spotting them can be very difficult. And then the things that we have observed, our observations are rather biased. So most of those are observed on the coast, about 30 to 50% of the observations. And overall, we only have records available for about 5% of the ocean volume. And when I say records, I'm talking about point observations. These are not necessarily time series. These aren't necessarily persistent observations. We have a sample from about 5% of the volume here. So this combination of things, the fact that the ocean is very large and very underobserved, combined with how dynamic it is, makes it extraordinarily difficult to do any sort of bioscience in the sea. So as we think about more densely sampling the sea, we have to start framing it as a big data problem. And if we're thinking about it specifically from the perspective of imaging, which, as you'll see in a moment, is a lot of what I've devoted my time working in this space on, you have to start thinking about how to work with that big data product. And when you combine these things, you can actually think about them either in terms of the biology and the physics, as I've listed out here. The ocean's dynamic and heterogeneous. It's large, it's biologically diverse and underobserved, and kind of translate that into some of these interesting machine learning problems that our computer scientist colleagues are thinking about with some regularity. Things like when you have a dynamic and heterogeneous environment, you're going to encounter what they refer to as distribution shifts. So that can be in terms of the organisms that are there, but also in terms of the camera system that you might be using, the way the environment appears in the images, that kind of thing. Because the ocean is large and sparsely populated, you're doing some form of anomaly detection. Most of what you're actually looking at, if you put a camera out there and are just sampling in a brute force manner, is empty. That diversity leads to long-tailed problems, and these are known to be very challenging for automated systems to deal with. And finally, that underobservedness of the deep sea, the fact that we don't know a lot of the stuff that we're actually looking at, means that it's an open set problem. So you have to have a system that knows what to do with new and novel objects that it might be encountering. But the sampling in the ocean has not been restricted to imaging, obviously, and in fact, we've had this long history of evolving tools to try and get us towards answering some of these biological questions that we have. I'm sure everyone here is very familiar with the different types of nets that we've put into the water over the years, going back a very, very long time. I love this image here. For those of you who aren't familiar with San Diego, this is a picture taken from the Scripps Pier at the turn of the 20th century, right around when Scripps was founded. And the area behind the gentleman with his net is La Jolla Shores, which is now one of the most expensive area codes in the United States and populated with fancy beach houses. And, there it's just a lovely cliff band. Things change. Of course, these sampling devices have gotten more complicated as we've started doing bottom trawls that sample in different ways and can get quite complicated. Like everyone's favorite behemoth of a sampling device, the Mach NestNet. And for those of you who are unfamiliar, that's the multi-opening and closing, I can never remember what the rest of the acronym stands for, that allows you to sample in a stratified way as you're going up the water column. And you can get some really, really wonderful data from these things. And in fact, you're going to be able to get genetic information. You're going to be able to actually put something under a microscope and get really, really firm species identification. But they're extremely labor intensive, very hard to deploy, and very hard to process the data. So as such, in the past 30 or 40 years, people have started looking more and more at using imaging systems to try and more densely sample in the sea. And these have been deployed in all sorts of different ways, from systems that sit in one place and image over a long period of time, like this baited underwater video system that just is a set of PVC pipes and a GoPro with some food attached to it, to things that are more complicated, like the camera tripod that is deployed out at Station M off the coast of California. So that thing sits at 4,000 meters and takes pictures every hour, I believe. There are other moored systems that look at much smaller organisms, like the Imaging Flow Cytobot, which pumps water into the system and takes pictures periodically. You have instruments that are towed behind vessels, like the In Situ Ichthyoplankton imaging system. And then, of course, concurrently, you have these camera systems that are then being deployed on autonomous vehicles to try to do things, sampling more densely, both temporally and spatially. And there are loads of these that have been deployed from all sorts of institutions like Woods Hole and the National Oceanography Center, and of course, where I'm currently working, the Monterey Bay Aquarium Research Institute. So combining these modalities, an autonomous vehicle with these camera systems, allows you to get new and different data than we were able to access before, but it creates a whole host of challenges with how to work with it. But it does allow us to access things at a huge range of body size scales, and in addition to those spatial temporal scales. So for example, you can start at the very, very tiny things. Some of these images might look familiar to some of you. Starting down at the individual cellular level on the scale of tens of microns, the same organisms that are the little discs can form into these big long chains. You can go up an order of magnitude and get to Fred's favorite, the copepods. And then, of course, you can go even larger than that and look at colonial organisms, big gelatinous things. And then going up yet again in size scale, you can get enormous gelatinous organisms that function in similar ways, like this very large siphonophore that we've seen off the coast of Central California. When we see all of these things, this big diversity of size, this big diversity of sampling modalities, you have to figure out what to do with all that data. So at a very high level, what people try to do generally in the marine ecological realm is some form of supervised learning. So for those of you who are unfamiliar with that process, this is a graphic that I adapted from a colleague's paper that came out last year in Annual Review of Marine Sciences, where you have your instrument, your acquisition system that can be either a lab instrument, it can be one of the many in situ systems that we were just talking about, where you collect a bunch of images, you do some amount of manual sorting to find a set of gold standard images that you then use to train some sort of automated model. And once that automated model is working to your satisfaction, the hope is that you can then take new unlabeled data that you're getting from those same systems, pump it through the model, and then have that output abundances, concentrations, biovolumes, that kind of thing that can be used for downstream analysis and modeling. So in an ideal world, you work on getting these images sorted and set up in a way that allows you to train a model that's then just going to work. Unfortunately, that's not typically what happens. And learning how to deal with that is quite challenging. But a bunch of people have tried this over the years and done all sorts of different flavors of this for many different types of marine ecology. People have been doing some form of supervised learning, that workflow that we were just describing, using hand-engineered features like this 2005 paper by Blaschko et al., where they looked at a whole bunch of different ways of using outlines of animals to try and sort images from FlowCam, I believe. People have attempted to do texture analysis to try to sort images of corals. And more recently, you have people trying to use these new parts-based analysis techniques to do behavioral studies by tracking the movement of individual portions of animals. And all of this is using some type of automated system under the hood, but it is doing that same larger framework of supervised learning, where you start with some data that you've collected, label it by hand, train a system, and then hope it's going to work for all the new data that's coming in. But again, that is something that happens in a narrow set of cases. When you're actually putting something out in the real world, your system will break, and figuring out how it breaks and how to fix that is a lot of what I've spent my research time thinking about, because it's both critical for getting to the interesting ecological problems that we're interested in, and also a very, very challenging engineering problem. So I first started encountering this in my personal work when I was at Scripps, working on the Scripps plankton camera system. So for those of you who are unfamiliar with Scripps, it's on the coast of Southern California. It's quite close to the Mexican border. So the pier, which is where I deployed this imaging system that I helped build and maintain, is right there where the X marks the spot on the treasure map. So zooming in, you might recognize the pier there. Well, I don't know. Actually, that's not even close to the image that I showed at the beginning. The image at the very beginning was from Scripps Pier. But Scripps Pier extends about a quarter of a mile, 250 meters off the coast. It is a tidal ecosystem, so we've got all sorts of influences from that type of physics. It's very dense. We have a lot of stuff in the water. Storms will kick up sand, that kind of thing. And if you zoom into the wide part of the pier, we built and deployed an imaging system that lived underneath the pier deck at a nominal depth of about 10 meters, again, tidally dependent, that consisted of 2 imaging systems, one that looked at things that are more in that tens to hundreds of microns in size, and the other one that looked at things in more of the millimeter to centimeter range. So they had different microscope objectives on them. They looked at different sample volumes, but they imaged in the same way. It was a dark field microscope system. So we had a dark background with light foreground pixels, so you could pretty easily segment out the images. And importantly, this is plugged in to a Ethernet cable that runs up to the pier deck and plugs directly into our servers. So we could see in real time what the population was doing underneath the pier. And this is a point measurement, but it still gets us this very, very dense temporal sample. And because we were able to look at it in real time, we were able to generate mosaics like this that we could look at and refresh on a web page to see what was out there. So this is from these are images from the higher resolution system. So you can see some serratium in there, that kind of thing. And as you might expect, we got lots of data from this. So I apologize that the axes on that timeline on the top are totally illegible. I have a different version of this slide where I zoom in, but trust me when I tell you that the x-axis is time. This is about 2 years worth of data that's being shown up here. The y-axis is counts, but it's on a logarithmic scale. So those peaks that you're seeing in those green lines are on the order of tens of millions of regions of interest a day. And those fluctuations that you're seeing are related to changes in the population, but also, when we have events that kick up sand or send dead kelp into the field of view, that kind of thing. And you can imagine what would be, the best case scenario here is that we have some sort of system that can sort these, because we're talking billions of regions of interest a year, way more than we could deal with by hand. But if you had some system that could process these, you could separate that timeline out into something species specific or functional group specific, and do some interesting ecology, thinking about all sorts of questions that you might ask about a coastal ecosystem like this. So one of the ones that we were targeting, and this was driven by my colleague at Scripps, Kasia Kennitz, was tracking harmful algal blooms. So we saw some potentially harmful algal bloom formers in this data, and thinking about how to track those in real time was a question that we were really interested in tackling. So there were 4 in particular that we were focusing on: Akashiwo, Chatanella, Dinophysis, and Lingliadinium. And these were things that we were seeing in that higher volume camera system. And then we went up a size scale to look at some of their predators. So in addition to thinking about the animals themselves and looking at some of the bulk signals that we were getting from other sampling measures, we used the bigger system to look at some of the grazers, ciliates and copepods. So as it turns out, when you're trying to actually work with this in real time, despite having classified many, many thousands of these images and trained a system that worked well in testing, as we actually deployed the system, it didn't work as well as we anticipated. So I'm going to walk through this plot that demonstrates some of that behavior. So on the x-axis here, we have date. This is about 2 years worth of data from July of 2015 to July of 2017. And on the y-axis, we have prevalence. So that's just the percent of data on any given day that is the organism of interest. So this is just looking at one organism at a time here. I'm going to show you data from 2 different outputs here. So the black triangles are manual counts. So these are the counts of our organism of interest in randomly selected 5-hour chunks of data that were not used for training purposes. So these represent new populations relative to the training distribution. So again, this is hand labeled. The human annotator went through those 5 hours of data, looked at everything that was in it. And then we sent our automated classifier that we trained, which was a ResNet 18 model, just very small, a rather simple system that's shown in these gray circles here. So as you can see, there are situations where it works somewhat well. It's relatively close to the human estimates. We did train it deliberately to have high recall at the cost of relatively low precision. And all that means is that we trained the system to be sure that we got all of the target organisms, even if it meant that we had a high number of false positives. So it would be easier for a human to go back in and check those if they could see, okay, there are a couple thousand of things that the computer thinks belong to my target, and I'm just going to find the ones that it got wrong, but be confident that it wasn't missing any of the things that it was interested in. But even then, it's not a consistent bias. So we have some situations where our estimates from our classifier were rather close, but then a couple, particularly in 2017, where the classifier's estimates are quite wrong, often by quite a large margin. And it turns out that in some of these situations, what we're seeing are the impact of new regions of interest, new things that we were imaging that did not show up in our training set. So in this particular set of days here that I've highlighted in the red box, we're seeing these bits of what we think are kelp fragments. So we had a big storm event. It was a bad year for kelp. They weren't especially healthy because the sea temperature was quite high. And we think these are bits of that material that were getting ripped up from the nearby kelp forests and drifting into our volume. And likewise, later on, we saw these sand grains that were getting kicked up by a different storm. So we had a big wave event. Sand particles from the bottom near the camera system were getting flushed up, and these were not well represented in our data. So what we took away from this was, I mean, we were then able to take this information, retrain our classifier, but every time we attempted to do this experiment, there was something new or different that was causing trouble. So we came up with a human in the loop error estimate system where we had a human expert go through the output of the target classes and check a small number of them to get a error estimate for the classifier in a particular day. So, at a particular time, you have this quantification of error that allows you to correct your population estimates for the purposes of the ecological studies. And so Kasia was able to use this to put together this really nice time series where she was then able to try to look at some of the drivers of this plankton population. So again, on the x-axis, we have time from January of 2018 to July of 2020. The y-axis in all of these is log abundance per day of the target species and the grazers. So the target species are in the top two panels and the grazers are in the bottom here. And I don't want to get too into the details of the ecological findings, but if you're interested, I would encourage you to check out her paper. She did some very nice analysis there that demonstrated what it was that was controlling the population of these algal bloom formers off the coast of San Diego there. But from a more engineering perspective, where does this leave us? So we have this huge volume of water, and we have all of these instruments that allow us to look at it in these cool, new, different ways, but we have these imperfect systems that are attempting to work with that data. So there's still work that needs to be done to operationalize these automated tools. And in my mind, the critical part of that is exposing and understanding those sources of error and how we think about what the best way of deploying these automated systems is. Is it really fully autonomously? How much do we need a human in the loop? What are the limits of where we can deploy these systems and really trust what's coming out the other end? And for the moment, I think that is going to be a trust but verify sort of process. As I've moved to MBARI, I've started thinking about the same type of procedure but in an autonomous framework, in a bandwidth-limited framework. So at Scripps, we had this instrument that was sitting off the pier that we had plugged in. We could always look at the data, but what happens if you're taking one of these imaging systems and putting it onto an autonomous vehicle and attempting to do something useful with it in the field? And I've been very lucky to have the opportunity to think about that problem quite a bit in the context of my position at MBARI. And a lot of that has been based on this new imaging system that was being put together right as I was showing up, which we're calling Chitin. And this is a picture of the instrument on a bench. So what Chitin is a 7-camera array. So all of those cameras put together gives you about 140 megapixels at about 50 microns a pixel. So it's looking, again, at things that are in the hundreds of microns to sort of several millimeters, maybe up to a centimeter in size. When you combine all of those camera volumes together, you get about 2 liters per frame. We're not necessarily thinking about it just from an increase in sample volume. We're also trying to do some 3D stuff with the different pairs of the systems in a stereoscopic way. And importantly, from my perspective as someone that likes to think about the computer science side of things, we're running the entire system on 2 of these pretty powerful NVIDIA embedded chips. So we have some computational horsepower on board that we can utilize to try and classify that data in real time. So this whole setup fits inside the nose cone of MBARI's long-range autonomous vehicle. So the system is very power efficient. It can run not with our camera system on it because our camera system is pretty power hungry, but it can run by itself for a number of weeks depending on what payload you put on it. When we have our camera system on there, we can run it for a little more than a week before the battery starts to complain, and then we have to bring it back and charge it up. And we put that all together. This is an image of the system in MBARI's test tank where we are doing some lab experiments before we put it out in the water. So you can see the illumination ring around it. The sample volume sits out front of the instrument itself, presumably in undisturbed fluid. So the way that we typically deploy this system is in a yo-yo pattern, which, again, I'm sure you all are familiar with, where you have your instrument sitting at the surface. You can communicate with it over satellite, or if you're close enough to shore, you can communicate with it over the cellular network, and you can program it to go to a certain target depth, return up to a shallow depth, and then repeat until the mission times out and it returns to the surface and checks in. So when we have chitin on there, it is running in a manner like this. So it's doing that yo-yo, it's imaging out front, and in real time, it does some amount of object detection and region of interest extraction. So again, this is an imaging system that has light foreground pixels on a dark background, so we can pretty easily get those objects out. You can then look at collections of those objects and send that information back to shore to get some information about what's happening in the community in real time. So in this plot, we have on the x-axis now, instead of time, we have longitude. So this is a spatial section. We have depth on the y-axis, so going down to about 300 meters. And the color scale here is the size spectral slope. So this is computed just based on the size of all of the regions of interest that we're seeing in the imaging system. But again, what we're doing here is very brute force. So we turn the camera system on, we set the frame rate, and we have it run these yo-yo patterns, and we can get something like this back. And this is undoubtedly useful, but it'd be really cool if we could actually update the vehicle behavior based on this signal that we're getting. And in fact, this is work that's already been happening using different instrumentation at MBARI. So this is an image from my colleague Yamu Zhang's paper a couple of years ago in Science Robotics that did persistent eDNA sampling in the deep chlorophyll maximum layer off the coast of Hawaii using a system of 3 instruments. So you have 2 of these long-range AUVs, OPA and AKU, and a wave glider on the surface, MOLA. So the wave glider on the surface is serving as a hub to kind of communicate between the 2 instruments. OPA is driving this corkscrew pattern to get information about the physics of the system. So it's locating and tracking that deep chlorophyll maximum using 1D CTD type signals, and then transmitting the location of the DCM to AKU via MOLA, and AKU's sitting there in the DCM taking eDNA samples as it goes. And using that workflow, they were able to sit in the DCM fully autonomously with this system of 3 instruments, I think for 8 hours. It may have been 10 hours. It's well worth checking out the paper. It's a very cool read. So we would love to do this with imaging. And I mean, doing it in the DCM would be great, but there are all sorts of other ways that you can imagine taking these pictures, updating the vehicle behavior that would really benefit from actually seeing what it is that's in front of your vehicle. So what would that look like? Schematically, the way that we think about this is we have a vehicle, we have our camera, and there's a user. And we have ways of communicating between these. We have a processing model and a camera vehicle interface that makes all these things talk to each other. So you, as the user, have some amount of control over this system. So say that you set a target organism that you want your processing model to pay attention to. When the instrument is out and running, the camera is collecting raw data and then sending that to storage for downstream analysis. But at the same time, the camera is sending that information to the processing model. The processing model then extracts some salient information. That could be, these bounding boxes, the little crops of the images that we're seeing, some summary information about the particle, about the particles, and then some information about whatever the particular target animal that the user designates. That information gets sent back to the camera vehicle interface, and then will trigger a behavior change on the vehicle. So schematically, again, what you have is your camera system that's taking pictures, sending it to the processing model that knows to look for a particular target, and then using some summary about that target to change what the vehicle itself is doing. So we've made some early experiments starting to get at trying to do this type of thing. And what we decided to focus on for the purposes of these experiments were krill. And part of this is driven by work from some of our collaborators at MBARI, who are really interested in blue whales and blue whale migrations. So they've established an observatory off the coast of Central California, as you can see here. So you have the Gulf of California and Baja down there at the bottom of the map. So we're about mid-coast there in that black box. So they have a hydrophone array that they've been deploying for quite some time and use that to track the whales acoustically. And what we did was we took one of these long-range autonomous vehicles with the chitin imaging system on it and deployed it coincidentally with the acoustic array to try and see if we could spot these krill as they were doing their acoustic surveys. So you can see in this image another map here, and the purple line that you can see in the bottom left corner there is the track of the autonomous vehicle as it's kind of crisscrossing back and forth over the shelf there near where the acoustic array is. And in this set of experiments, we did in fact see lots of krill. So these are an array, or sorry, a mosaic of images of the krill as seen by chitin. So they're long, they're skinny, they're bright. Some of them you can see the eyes, but they're very distinct relative to lots of the other particles that we see. So before even bringing in any fancy machine learning models, we wanted to just test this out using morphological features, so things that we could measure directly from the image. So what we came up with was a way of separating the regions of interest that we're seeing just based on the length. We're getting some estimate of how elongated the particle is and how bright it is, and then using that to make a count of the number of elongated objects or krill-like objects that we're seeing in the frame. And as we were running this in real time, we were able to actually observe some of the shifts in the krill population over the course of the day. So these plots that we're looking at are actually what we see in the dashboard. So this is information that's getting transmitted back via satellite in real time. On the x-axis, we now have time. So this is one 24-hour period on the 19th of November of last year. The top panel, and I'm sorry, the y-axis is depth. The color scale is what differs between these two plots. So on the top, we have particle counts. So this is just everything that we're seeing in the camera system mashed together and put on this color scale here. And then on the bottom plot, we have the count of the elongated particles. And you can see that in the top, we're looking at thousands of counts. And in the bottom panel, we're looking at tens of counts. But you can see that there are these hot spots of these elongated particles that we were able to detect using this workflow. And they don't necessarily line up with where the hot spots of just the raw particle counts are. So we're confident that we're seeing something representative of the actual particles. And we could, in fact, confirm this when we brought the whole system back to shore. So if we zoom in just on this one upcast of one of our yo-yos and look at the video output, we can spot where these bright patches are and actually see that they are, in fact, these long, skinny things that we wanted to detect. So I'm gonna play a video in just a second here, but to orient you as to what you're looking at, these red boxes here are detected particles that are very, very small. So we weren't saving those regions of interest, but they did contribute to the particle count. The green boxes are things that were big enough to be saved as regions of interest. And then the white boxes are things that the camera system thought were krill. So and the white boxes are being put on there in real time. So this is the output of the system right after it came back to shore, and we were able to look at the full frame video. And then on the top, you have time in the middle and then depth in the upper left corner there. So we're starting at the bottom of the profile at about 200 meters. So we hit the bottom of our profile at about 200 meters and we start coming back up, and we're seeing lots of our red tiny things and the green stuff that might be interesting, but is not the actual target of this experiment. And as we start getting closer to the surface, or not to the surface, but up towards 150 meters, we start seeing more and more of our krill friends. And eventually, as we get further and further up, it starts to get rather dense. And if we can measure this change in population, this spike in the number of krill that we're seeing consistently and confidently, then we could say, okay, vehicle number 2, come in and do some different sampling, or vehicle number 1, change what it is that you're trying to do. And schematically, what that might look like on a single vehicle is you have your vehicle at the surface again. It starts doing its, or rather, there's a population of krill or some other organism that you're interested in. Your vehicle starts its dive. It does a yo-yo or two. It detects that it is in a dense region and attempts to stop and sample it more consistently. After some period of time, it either loses that patch or it times out of its mission and continues along the rest of its course until eventually it returns to the surface. It would be great if this operated entirely autonomously. But again, something that we're really interested in doing is trying to figure out how to make that happen in a way that keeps the user in the loop. So if we return to our diagram that we see that I showed a little earlier, that if we take that and make it more supervised, what we'd love to do is be able to transmit information from the vehicle that gives the user some sense of what it's looking at and whether we can be confident in the behavior change that the camera is triggering. And we've also been able to demonstrate this as part of a different project where we can send images acoustically back to the surface using a situation similar to the graphic that I showed from Jan Wu's paper, where we have a wave glider at the surface with an acoustic modem, and we can send those target pixels back. My colleague Ivan Masinita published a paper about this quite recently, where we were actually able to send these images back to the surface, and then from the surface profile, send it back to shore. So we get these emails periodically with an attachment with a couple of representative frames of what we're looking at. So there's still a lot of work to be done to figure out how to do this in a statistically relevant way, where we can say that, okay, this set of like 3 regions of interest that we're getting back are representative enough that we can confirm or reject the behavior update that the system came up with. So this type of sampling regime could open up some really interesting ways of putting things out in the ocean. And there are lots of folks that are attempting to do this, not just us. We get these huge mosaics of images from something like chitin, but there are also all sorts of other instruments going out there, like the UVP that is going to go out on bioargo floats that will likewise get huge amounts of image volume back. And whether or not these can make a behavior change, being able to transmit information confidently is a major hurdle to getting to do the science with these things. And it's not just with these types of systems. We can also think about this from a larger macroscopic, if we're looking at bigger things. So we can do situations or systems rather, where instead of actually doing counts, you might want to track something. So you spot an individual and you try to follow it in real time. This is an example of that sort of behavior from a remotely operated vehicle that's carrying a stereoscopic imaging system that's running a model under the hood that's doing detection, finding the objects and locating them in 3D space, and actually driving the ROV to try and track it. So again, this is just for demonstration on a remotely operated vehicle. So the human is fully in the loop because it's connected by a fiber, and you can actually make sure that it's doing the right thing. But as the model detects the object that it's trying to spot, so the target here is Bathocordeus, which is a type of larvation. It finds it, acquires it, and then sets up the object track. and then just sits on it and images it for as long as the mission is set up to do. So being able to take this off of an ROV and put it onto an autonomous vehicle opens up a whole different regime of potential deployments and imaging routines that you could do to try and adaptively sample in a targeted and concentrated way to try and do a not better job, but a denser job of finding things and densely sampling them in the deep sea. Where do we go from here? In my opinion, we need automation to fully explore and monitor our seas, but while we're getting closer to being able to do that with these imaging systems, there does remain a good amount of work to do. And a lot of that entails development at this sampling and machine learning interface. So it's not just building the cameras or the models, it's putting them together in an interesting and constructive way that builds on the capabilities of both. Doing them in isolation just leads to situations where you're not really maximizing the potential of either of them. And from both of those perspectives, being sure that this is calibrated and making sure that there is supervision in the loop will be very important for the immediate future. And finally, data sharing is a very, very important aspect of all this. So having information in one place and being able to test it on another is critical to making all of this stuff function as smoothly and efficiently as possible. So with that, I know I've gone a little beyond the 45-minute mark. I want to make sure there's time for questions. So I'll just briefly acknowledge the lots and lots of people who made this work possible. Clearly, I didn't do all of this on my own. I'd particularly like to acknowledge Kasha Kennetz at UCSD, Kakani Katiha, and Paul Roberts at MBARI, and all the wonderful places that have given us money to do our cool work. And with that, I will thank you for your time and attention, and I'm happy to take any questions, either from the in-person audience or from anyone online. Thank you.
Hi, thanks for the presentation. A technical question first, and then a second one after. What's the resolution of the rate of the image getting output by your AUV camera array? And the second question would be, do you think there's a bias, let's say your krill experiment or measurement of those krill being scared away by these huge lights, like you could get away from the camera focus and then you're missing out?
Yes. So to answer your first question, we were running in that experiment, I believe, at 10 hertz, so 10 frames a second. The processing can keep up at faster frame rates. I can't remember what the maximum is that we've tried and still been able to keep up with the processing routine. But on the order of 10 to 20 frames a second. To your second question, yes, we're absolutely concerned about avoidance, particularly if you imagine stopping in your layer and trying to just drift and see what's there. We are building a red light illumination array to try and ameliorate that problem. And ideally, we'll be able to switch between those two illumination modalities on the same set of strobes, so we don't have to swap them back and forth. But the white lights were easier to acquire and put together, so that was the first cut. But yes, I mean, that will be very important, certainly for larger, more motile organisms like krill. Of course.
Are there any questions online?
Are your automated imaging tools often coupled with the autonomous eDNA sampling system developed by MBARI? Did everyone hear me when I read that out? I think it showed up on the video here. So not yet, but that is a big push at the institution. So there's another plankton imaging system that's just come online, and then there are a couple of instruments that look at or imaging systems that look at larger animals that are also going on autonomous systems, and trying to figure out how to make those things work together is something that's talked about a lot around MBARI. And given the capabilities that have been developed there with the ESP, the Environmental Sample Processor, I think is what that acronym stands for, that does fit on these autonomous vehicles, there's a whole lot of potential there. But in answer to the question, no, it hasn't been done yet, but it is something that I believe is in the development pipeline. And I don't want to speak for everybody at MBARI, but certainly in conversations that I've had around the institution, there's a lot of interest there.
So first, I'm totally sold out to this approach. I think it's, for me, it's the same kind of change than the satellite imagery went into right 40 years ago. It's absolutely nature. Technically speaking, how much of the whole process is dependent on a specific optical device, or like could you swap the VP6 for the Eton or the IZID or whatever?
Yes, absolutely. So the part of the reason that in that schematic that I showed back here, that I didn't necessarily put, chitin into that green box there where it says camera, is because in principle you could do this with any camera system and any type of target. So you would need to use a different model. You couldn't use the same processing model that we are working with on chitin for doing this. But having that same type of routine where you're counting objects based on some classifier, and then coming up with an estimator that allows you to make a decision on the vehicle is absolutely something you can do with a different imaging system. And I don't make any claims to chitin being the best way of doing this either. I think that it allows you to access interesting facets of the community, but that's true of lots of imaging systems as well. So I could readily imagine situations where you're using something like UVP or ISIS to do this same type of adaptive sampling with a different design. But the general workflow could certainly be utilized.
And I think another point I'd like to make is more comments, but maybe you can react on that, is from the list of people you wanted to thank for all that, it's definitely a team effort, and it's definitely so there is this technical and computer science aspect and developing these interfaces, etc. But is there any plans to have that at some point as a point of service for the larger community of researchers who don't have access to this team of this team of developers? And, that's open. So you say it's important to develop together the devices and the machine learning and get this interface, but it's the toughest spot for most teams to find people who are able to do these at the interface and to build a team and keep it. And it's very challenging.
Yeah, it's hard, and it's hard to develop the shared language that allows people to do that constructively. I think that we are at a point now where there are starting to be programs designed to educate people at that exact interface. So I think, 10, 15 years from now, there's going to be a whole crop of students coming up that are focused on that really think about things like machine learning for conservation or artificial intelligence for biological adaptive sampling, that kind of thing. So it will remain obvious, like a highly specialized thing, but I think it will be less uncommon soon than it is now. And to your comment about sharing and making this stuff available, I'm a big proponent of open source, but just because you make your code base open source doesn't mean that it's necessarily accessible. So coming up with ways to do that in a broader way is also a big challenge. And I don't have a good solution for that off the top of my head, especially when you're talking about running bespoke, expensive devices. These aren't exactly off-the-shelf components, but figuring out how to do that in a more broadly applicable way is an important step for future work, I would say. There's still a lot of development that needs to happen to make these things operational. I don't think this type of tool and this type of workflow is at a state where you could just kind of, give it to a new team, spend a week teaching them how to use it, and then letting it go off and do its thing and expect to get good science out of it. A laudable goal, but not necessarily one that we have all figured out at the moment.
I think it's over now. So
yes,
thanks a lot, Eric. And
yeah, thank you all for having me. This has been fun. Thank you all for listening online.